versus secondary, okay? So primary versus secondary immune deficiency disorders or diseases. And the big difference between the two is that primary is really congenital, something that you're born with. Okay? It's coded at the level of the DNA, and during development, something takes place, and I'll show you how that manifests. And then secondary, another word for that is going to be acquired. You catch it, you pick it up somewhere. Now, acquired could be acquired in utero, right? So an example of a secondary acquired is like HIV. We're going to talk about that example. But HIV isn't congenital, right? It's not coded for, it's acquired. It's picked up, but it can be picked up during development, but that doesn't mean that it was coded by the DNA. So if we look at some examples of congenital, the tree that we're going to go through today is shown on this slide. So these are primary immune deficiency diseases, or congenital. They're single gene defects that are coded. But we've got really 5 different types in the field. B cell, T cell, combo, or both of them, complement and phagocytes. We're going to only talk about the first 3. But knowing what you know now, you can probably make some guesses as to what happens with complement and phagocytic primary immune deficiencies. But if you look at this tree, here is a pluripotent stem cell at the top. That pluripotent stem cell becomes a common lymphoid precursor, and it either goes down a tree making T cells or down a tree making B cells. You with me? Ignore the red squiggly lines and all the boxes with the $50 terms in it for now. We'll get to some of them. So your bone marrow is this box right here. This is all happening in the bone marrow, where you have stem cells in the bone marrow. That's that pink box. And that stem cell either is going to move out and move into a pro-T, go to the thymus, or it's going to go down the B pathway, and it manufactures IgM, G, A, and E, your different varieties of antibodies that come from B cells or plasma cells. Okay? So we're going to look at some of the little disorders that take place along this tree. So this is a good study tool. So the first one we're going to blow up and look at, is right here. So this is the upper left part of that box where we've got in the bone marrow, we're here, we're going to look at this deletion that takes place, referred to as X-linked Agammaglobulinemia, known as Bruton's disorder or Bruton's disease. So in Bruton's disease, it's sex-linked. It's a rare X-linked genetic disorder that was discovered in the early 1950s. It affects the body's ability to fight infection because the body doesn't make any mature B cells. Right? So if you pause right here, you only make pre-B cells. If I back up one slide, that's this one right here. You don't make any of the mature B cells, so you don't make any of the antibodies. You only make pre-B cells, but they don't differentiate. The problem is that the heavy chain is synthesized, but the light chain is not, so it's incomplete. The signs and the symptoms in patients are either low or absent B cells. They have low or absent plasma cells, underdeveloped lymph nodes, but they actually have a very normal T cell response. Most viral and fungal or protozoan infections are fine because the T cells can still fight off those things. But they have a lot of problems fighting different types of bacteria. So they have chronic infections that are present. A lot of times bronchitis and pneumonia is taking the patient. So this is our X-linked Agammaglobulinemia, or Bruton disease. If we move to the next one, this is known as a selective IgA deficiency. The selective IgA deficiency, by name you can see, it only prevents you from making your IgA, but you still make your IgM, your IgG, and your IgE. So you have a muted antibody production response, but it's not completely absent. So you get a lot of infections where IgA is typically found. It's found in the blood and on body secretions. This one is the most common primary immunodeficiency disorder that we see. All right, so the next one up is T cells. So if we go to the other side, we're going to look at the DeGeorge syndrome. This one right here. So the DeGeorge syndrome was described by pediatric endocrinologist Angela DeGeorge. And these patients have a partial or a complete lack of thymus. So they don't make any mature T cells. They're very prone to viral, fungal, or protozoan infections. They typically can fight bacterial infections just fine because they make a normal B cell. DeGeorge syndrome. Again, not nearly as common. Again, the most common being the IgA selective deficiency. So these are all patients that are born with these kinds of things. Now, the last one that we're going to look at that's a combo is SCID, severe combined immunodeficiency. Another name for it is the ADA deficiency, adenosine deaminase deficiency. So if you look at the very top, this is the pluripotent stem cell, this is the bone marrow. You make the precursor, but you don't make any pro-B or pro-T cells. This is the ADA deficiency. So these patients don't make any B or T lymphocytes. So in these patients, the entire immune system is affected. They're at significant risk for just about everything. There was a individual, a young boy in the 1980s, before most of you were born, named David Vedder, and he was affectionately referred to as Bubble Boy. And David Vedder suffered from this ADA deficiency, and he lived within a bubble within the hospital setting, and he wasn't able to have any human contact whatsoever. And he was one of the first popularized ADA deficiencies that we saw in sort of the public media. Okay, now we're shifting from primary to secondary. Now, secondary, I'm only going to show one example. I only showed two examples of B cells, right? One example of T cell, and then a combo, the SCID. And now I'm only going to show one example of secondary. And that's all you have to know for the example. Okay? But I want you to be familiar with the concept that there are many different types of secondary immunodeficiency disorders. They could be from malnutrition. Like a patient could have a zinc deficiency or an iron deficiency. They could be malnourished in other areas and their immune system is suppressed. That would be considered a secondary immunodeficiency disorder. Okay, diet. Those of you that are into diet, this is kind of a biggie. Stress. We were just talking at the break about stress as a stimulant for triggering autoimmune disorders. So a lot of patients, some of you have been asking, okay, well, I used the funnel cake as an example, right, of this type 1 diabetic family friend. But a lot of times, stress, where glucocorticoid levels go up, that ramps up or can compromise, in some cases, the immune system, where your body's under attack. Cancer itself can cause attack on the immune system, especially if it's at the level of the bone marrow. And then iatrogenetic causes. So like these causes are usually, the term iatrogenetic means a side effect or indirectly as a result of. And so these could be complications due to a medical treatment or medical therapy that now causes a secondary side effect of suppressing the immune system. That would be secondary. Or even infection. In an infection response, your body is fighting an infection, and so it's almost like if it's fighting an infection, it's taking soldiers from everywhere else and putting them over here. And then there are areas that are at risk of being weak. So some of you have experienced this yourselves or with friends. It seems like they kind of get sick after sick after sick. They can't get over it. And there's stress involved there. There's a lack of sleep, and then there's infection. And that can be a secondary side effect. But the one that I want to highlight in our remaining time together, I'm definitely not an expert on. But it's one that obviously has gained a lot of public attention since the 1980s, and that's HIV. So HIV, to our best guess, likely arose from a simian immunodeficiency virus in the 1930s and began diversifying. It was first found in humans in 1959 in the Democratic Republic of Congo. Today, heterosexual activity is the most common transfer name. Heterosexual activity. The transmission can be through blood, intravenous drug use, sexual intercourse, maternal fetal transfer. There have been accusations that it's carried in the saliva, but there has been no confirmation of solid concrete nature that that's a real risk factor. That being said, I think a lot of universal precautions are taken, especially in medical situations. The disease, the virus itself leads to a disease that we're going to talk about. And I just tripped myself up on that. But the virus itself, there's two main types. There's HIV-1, which is more common. And it's usually isolated, or it has been isolated for patients in the US, Europe, and Central Africa. And HIV-2 is the one that's more common in Western Africa. The structure of it is it's a retrovirus. It has a lipid envelope or an outer casing with a virus core. The lipid envelope comes from the host. And it has an aggressive mutation rate, which is one of the reasons it's been so difficult to actually treat or even cure. The pathogenesis of HIV, or how it comes about within the body, is by targeting the immune system specifically. So students tend to focus on HIV as the only example of a secondary or acquired immunodeficiency issue, and they forget for the exam that we have things like diet and stress and infection and all these other things that are far more common. Okay? But this is kind of the big, the big one, the big exclamation point. So you can kind of see on this chart, this cartoon diagram, this is the cytoplasm of the cell, and the cell that we're talking about is a what? It's expressing Cd4, so what kind of cell is it? It's a helper T cell. That, unfortunately, is the cell it likes to attack. It's the cell of choice, is the helper T cell. And the helper T cell is so important in stimulating a number of different immune responses. It's kind of what we call the linchpin of the immune system complex or mechanistic pathways, where the helper T cell can stimulate cytotoxic T lymphocytes, but it also can stimulate an antibody response. And so if you take out the helper T cell, you kind of kill the most important player. So this is a cytotoxic T cell. It's not the only cell type that can be infected, but it's the main one. So dendritic cells and macrophages also can be infected. And in the central nervous system, we can actually see meningitis-like symptoms as a result of HIV attacking in the central nervous system, leading to what we refer to as an AIDS dementia complex in these patients. So what we have here is we've got our HIV virus, and again, it's a retrovirus. It has these two important docking proteins, GP41 and GP20. So the GP20 is what binds to the CD4. It has an affinity for the CD4. But in order for it to get there, it has to go through this conformational change. And in order to facilitate that docking, it needs what's referred to as a CCR5 receptor. So the CCR5 receptor is essential for that GP20 to bind to the CD4. And actually, believe it or not, there are patients that have been identified to be CCR5 recessive. They lack the CCR5 receptor, and they are actually immune to HIV. They've been exposed to the virus, and the virus cannot infect their CD4 cells. So if this continues, right, CCR5 receptor is present, you get docking, you get penetration of the membrane, and then you get fusion, and it injects its genetic information into the helper T cell. Now, the progression of the infection of the virus goes through a number of different phases. And most oftentimes, during this clinical latency phase, the clinical latency phase is where you've got infection. Viremia is when you have the virus circulating around in the bloodstream. And then clinical latency is where it kind of remains in a at-check status. So a lot of times in clinical latency, only about 0.1% of the CD4 cells, or the helper T cells in the organism, are actually infected with the virus. And it kind of remains dormant in that period of time, and it could be for years. It could be as long as 10, 12, 15, maybe even 20 years they could be in clinical latency. But at some point in time, it comes out of clinical latency, and you get this excessive viral replication. It's not super clear right now what causes this sort of awakening to take place. And then you get the destruction of the remaining CD4 T cells. And when you have the destruction of the remaining CD4 T cells, that's when we have our problems associated with AIDS. So in this graph, we actually take a look at a number of different parameters. There's basically 3 axes that I want you to look at. The first axis that's over here on our classic y-axis is our Cd4 cell population. And it's indicated by the blue dotted line. So you can see we've got about 1,000 cells per cubic millimeter volume of blood. And this primary infection is going to occur when the virus actually is inserted into our first CD4 cell. And that primary infection drops it to about half, right? There's some little rebound that takes place, but essentially during this clinical latency period, we have about half the T helper cells that we normally would have. Now, the second axis that I want you to look at is over here on the far right, and this is the viremia scale. So this is the yellow dotted line that's giving you the number of RNA copies per milliliter of blood plasma. And so you can see there's this huge spike in the first 6 weeks of the infection. Then it kind of drops off, and it kind of remains somewhat flat during the clinical latency. And these sort of like hash marks that are here means that it's not to scale. It's not a continuum of time it's actually drawn to scale. But at some point in time, the third axis is the x-axis, which is time. and here's that little break. At some point in time, years later, this graph is showing about 6 to 7 years later, the virus reaches a point where it replicates fast enough, and subsequent death of helper T cells brings this into what we call the crisis mode of the disease. So we've got acute, chronic, and the crisis mode. In the crisis mode is where you've got opportunistic diseases that take over. And so patients usually don't die from AIDS. They die from something that they caught while they were immunodeficient. They die from pneumonia, or they die from the flu, or cancer shows up in these patients, and they don't have an immune system to fight it. Okay? So this is kind of a nice summary of what goes on within one example, HIV, leading to acquired immunodeficiency syndrome, AIDS. But it could be years later before it changes or switches over from the virus to the manifestation of an immune deficiency disorder, which would be a secondary or an acquired disorder. Okay? So I want you to be able to compare and contrast congenital versus acquired. Any questions? Okay, we're going to finish like 2 minutes early. Merry Christmas. Early.